Hello, hi. In this presentation, let us look into some of the important molecular techniques like blotting techniques. What is blotting? Blots are techniques for transferring DNA, RNA, and proteins onto a carrier so they can be separated and often follows the use of a gel electrophoresis. So the southern blot is used for transferring DNA, the northern blot for RNA, and the western blot for protein. So let us look at the types of the blotting techniques. The first one is southern blotting. It was Sir Edwin Southern, the professor of biochemistry and fellow of Trinity, developed this particular method in 1975. He developed this procedure at Edinburgh University more than 20 years back. The method involves the separation, transfer, and hybridization. The method is routinely used for molecular biology for detecting detection of a DNA sequence in DNA sample. The DNA detected can be single gene or it can be part of a larger piece of DNA such as viral genome. And southern blotting combines with agarose gel electrophoresis for size separation of DNA with methods to transfer the size of separated DNA to a filtered membrane for probe hybridization. The principle that in this particular southern blotting, the mixture of molecule is separated. The molecules are immobilized on a matrix. The probe is added to the matrix to bind the molecules. Any unbound probes are then washed and removed, and the place where the probe is combined corresponds to the location of the immobilized target molecules. And these are all the operators that are used for southern blotting technique. These are called electrophoretic unit. You can see the dye glass apparatus. And the steps involved in blotting techniques, southern blotting techniques, are first we need to digest the DNA with an appropriate restriction endonuclease. And you know that the complex mixture of these fragments subjected to gel electrophoresis to separate the fragments according to their size. And the restriction fragment present in the gel are denatured with alkali and transferred onto a nitrocellulose fiber or nylon membrane by blotting method. And this procedure preserves the distribution of the fragments in the gel, creating a replica of the gel in the filter. And the filter is incubated under hybridization conditions with a specific radio-labeled DNA probe. The probe hybridized to the complementary DNA restriction fragments and then pair. And excess probes are washed away and the probe bound to the filter is detected by autoradiography, which reveals the DNA fragment to which the probe are hybridized. And these are all the stages of southern blotting techniques and the separated fragments which are visible in the radiogram. And the application of these southern blots are used in gene discovery and mapping, evolution and developmental studies, diagnosis and forensic sciences for preparation of RFLP maps, and identification of the transferred gene in transgenic individual. And southern blots allow investigator to determine the molecular weight of a restriction fragment and measures relative amount of the different samples. It is also used to detect the presence of particular bit of DNA in a sample. And it's also analyzed the genetic pattern which appear in the parent DNA. So another one is northern blotting. Northern blotting is a technique for detection of a specific RNA sequence in the pool, and it was discovered by James Alvin and George Starr at University of Stanford, and was named such by analogy to that of the southern blotting. Southern blots, and you can see steps involved in northern blotting. RNA is isolated from several biological sample, and then RNA is more susceptible to degradation than DNA. So sample are loaded on gel, and RNA samples are separated according to their size on agarose gel. The resulting gel following after the electrophoresis run, the gel is then blotted on a nylon membrane or nitrocellulose filter paper by creating the sandwich arrangement. The membrane is placed on a dish containing hybridization buffer with a labeled probe. Thus, it will hybridize to the DNA on the blot that corresponds to the sequence of interest. The membrane is washed to remove the unbounded probes. The labeled probe is detected using autoradiography in similar manner as that of the southern blotting. The application of this northern blotting it is used to study gene expression at a level of mRNA and detection of mRNA transcript size, and used to study the RNA degradation, and used to study RNA splicing and half-life of the RNA. Disadvantages for southern blotting. The northern blot, sorry, the disadvantage of northern blotting, and it is a method relatively less sensitive than that of the nuclease protein assay and as well as RT-PCR. Detection with multiple probes is a big problem in this case, and if RNA samples are even slightly degraded by RNAs, the quality of the data and quantification of the expression is quite a little bit tough task in this case. And the last one is Western blotting, and it is a immunoblotting techniques which rely on the specificity of binding between a protein of interest and a probe, that is antibody raised against that particular protein, and then it allow the detection of a protein of interest in a mixture of many others similar molecules. And the steps involved in this particular Western blotting: a protein sample is subjected to electrophoresis on the STS page. Electroblotting transfer the separated proteins from the gel to the surface of nitrocellulose membrane. The blot is incubated with a generic proteins such as milk proteins or BSA, which binds to any remaining sticky places on the nitrocellulose, An antibody that is specific for the protein of interest is added to the nitrocellulose sheet and reacts with the antigen. Only the bound containing the protein of interest binds the antibody, forming a layer of antibody molecule. After washing for removal of non-specifically bounded antibody, second antibody is added. It specifically recognizes the F3 FC domain of the primary antibody and binds to it. Antibody 2 is radioactively labeled and is covalently linked to a reporter enzymes which allow to visualize the protein AB1 AB2 complex. The application of this Western blotting in the confirmatory of HIV test, Western blot is also used as a definitive test for bovine spongiform encephalopathy. Some form of Lyme disease testing can also be done by using this technique.